The last section of your lab report is your conclusions or summary section. This section serves to highlight your critical findings. First and foremost, restate your key findings such as yields, chemical formulas, or concentrations as shown to the right. Make sure your conclusions are consistent with your results. If possible, it's best to relate your conclusions to the purpose of the experiment as shown to the right. If the purpose of the experiment was to find the unknown concentration of solution, then preface your results by saying that. Don't try to oversell what you've done either. Don't claim your results have major impacts or implications unless they really do. Simply state what you found in the context of the purpose of the experiment. Do not copy sentences from other sections. It is much better to restate your results and copy them from the results section. This section is to serve as a summary, not a retelling. Also, try to show that you understand your results. How precise is the answer? How precise could the answer be? If an experiment has multiple parts, how do the results of one part affect the results of the next part? This is shown in this example conclusion section by reporting the standard deviation of the different concentrations. Also, if you had any expected results or large errors, be sure to explain why. Which leads into the second and major component of the conclusion section. The second part of your conclusion section is the error analysis, which is where you will demonstrate your understanding of experimental error. The summary section should quote data from the literature, such as accepted or theoretical values, so that the audience or the reader knows what you should have gotten. You should explain significant deviations away from expected results, particularly those that are beyond 10% of the expected value. For example, if you have a large or small yield, what experimentally caused this? If you had errors, why were they so big or so small? Big errors are usually easier to discuss, but for small errors, you should still address what's causing you to be off. How could you prevent those errors in the future? When discussing sources of error, make sure your explanation is consistent with what you're seeing. If you have an overly high yield, attributing your error to factors such as losing material in a filter or spilling the sample doesn't explain the high yield, so don't mention them unless there is a reason to. If you want to state that factors caused you to get a lower yield so that the high yield should have been even higher, that's permissible. But don't discuss sources of error if they aren't pertinent to what you're seeing. It comes across to you, professor, that you hadn't actually thought through what could have happened during the experiment. If you omit a data point in your calculations, justify this in your error analysis section. Suspicious data points are a good way to highlight sources of error. And finally, make sure you explain the significance of percent error and standard deviation calculations. Were they large or small? What does the standard deviation tell you about the precision of your measurement?